Hi, good afternoon. Welcome back to New Age Cosmetic White EdTech Healthcare Consultancy. Most welcome to our daily lectures. Good afternoon to all of you. I am Dr. Bobby KORAH. We're on Chapter 6 of Molecular Biotechnology, titled Manipulation of Gene Expressions in Prokaryotes. The topics included are regulatable promoters, large-scale systems, increasing protein production, expression in microorganisms, fusion proteins, uses of fusion proteins, cleavage of fusion proteins, and surface display, and translation expression vectors, increasing protein stability, intrinsic protein stability, facilitating protein stability and protein folding, co-expression strategies, and then overcoming oxygen limitations, uses of protease deficient host strains and bacterial hemoglobin, limiting biofilm formation, DNA integration into the host chromosome, removing selectable marker genes, increasing secretion into the periplasm, secretion into the medium, metabolic load, summary, and review questions. The primary objective of gene cloning for biotechnological applications is the expression of the cloned gene in a selective host organism. Unfortunately, the insertion of a gene into a cloning vector does not necessarily ensure that it will be successfully expressed. Moreover, for many commercial purposes, a higher rate of production of the protein encoded by the cloned gene is required. In response to the need for a high expression rate, many specialized expression vectors have been created that provide genetic elements for controlling transcription, translation, protein stability, and secretion of the product of the cloned gene from the host cell. The molecular biological features that have been manipulated to modulate gene expression include the promoter and transcription terminator sequences, the strength of the ribosome binding site, the number of copies of the cloned gene, and whether the gene is plasmid-borne or integrated into the genome of the host cell, the final cellular location of the synthesized foreign protein, the efficiency of translation in the host organism, and the intrinsic stability within the host cell of the protein encoded by the cloned gene. There is no single strategy for obtaining maximal expression of every cloned gene. Consideration of the distinctive features of a cloned sequence is usually required before an optimal level of expression is found. The level of foreign gene expression also depends on the host organism. Currently, although a wide range of both prokaryotic and eukaryotic organisms can express foreign genes, many of the commercially important proteins produced by recombinant DNA technology are synthesized in E. coli. The extensive use of E. coli is understandable in light of the vast amount of research that has been carried out on its genetics, molecular biology, biochemistry, and physiology. Moreover, foreign proteins can usually be produced by this organism rapidly and inexpensively. However, other host systems such as Bacillus subtilis, yeast, fungi, and animal plant and insect cells are used to express certain cloned genes. Nevertheless, the strategies that have been elaborated for E. coli, in principle, are applicable to all systems. Gene expression from strong and regulatable promoters. The minimal requirement for an effective gene expression system is the presence of a strong and regulatable promoter sequence upstream from a cloned gene. A strong promoter is one that has high affinity for RNA polymerase, with the consequence that the adjacent downstream region is frequently transcribed. The ability to regulate a promoter enables the cell and the researcher to control the extent of transcription in a precise manner. The promoter from the well-studied lactose operon of E. coli has been used extensively for expressing clone genes. However, other promoters have distinct properties that make them useful for controlling expression. Thus, many different promoters have been isolated from a range of organisms. It might be seen that a good way to optimize the expression of a clone gene would be to insert it into a plasmid under the control of a continuously activated strong promoter. However, a high level of continuous expression of a clone gene is often detrimental to the host cell because it creates an energy drain, thereby impairing essential host cell functions. In addition, all or a portion of the plasmid carrying a continuously constitutively expressed clone gene may be lost after several division cycles, since cells without a plasmid grow faster and eventually take over the culture. Such plasmid instability is a major problem that may prevent the efficient production of a plasmid-borne gene product on a large scale. To overcome this drawback, it is desirable to control transcription in such a way that a clone gene is expressed only at a specific stage in the host cell growth cycle and only for a specified duration. This objective is achieved by using a strong regulatable promoter. The plasmids constructed to accomplish this task are called expression vectors. The next will be regulatable promoters. The most widely used regulatable promoters are those from the E. coli lac and TRP tryptophan operons. The attack promoter, which is constructed from the 10 region, that is the 10 nucleotide pairs upstream from the site of initiation of transcription of the lac promoter, and the 35 region of the TRP promoter. The left are PL promoter from bacteriophage lambda and the gene 10 promoter from bacteriophage D7. Each of these promoters interacts with regulatory proteins, example repressors or inducers, which provide a control label switch for either turning on or turning off specific transcription of adjacent clone genes. In addition, each of these promoters is recognized by the major form of the E. coli RNA polymerase holoenzyme. This holoenzyme is formed when a protein called sigma factor combines with the core proteins of RNA polymerase. Sigma factor directs the binding of the holoenzyme to promoter regions on the DNA. In the absence of lactose in the growth medium, the E. coli lac promoter is repressed, turned off by the lac repressor protein, which prevents the lac operon from being transcribed as shown in figure 6.1. Induction turning on of the lac promoter is achieved by the addition of either lactose or isopropyl beta d-thiogalactopyranoside IPTG. Let me spell that again for you. Isopropyl d-thiogalactopyranoside IPTG, a synthetic inducer to the medium. 6.2. Either of these substances prevents the lac repressor from binding to the lac operator, thereby enabling transcription to occur. In practice, lactose must be converted to allolactose by low levels of beta-galactosidase that are synthesized when the system is repressed before it can act as an inducer. The enzyme beta-galactosidase is encoded by the lac Z gene of the lac operon, and it is primarily involved in the cleavage of lactose into glucose and galactose. Transcription from the lactose promoter is also regulated by the binding of the catabolite activator protein, CAP, also sometimes referred to as the cyclic AMP, CAMP repressor protein, or CRP, to a region of the DNA. The cap box just upstream of the promoter gene, figure 6.1, when cap binds to the cap box, it increases the affinity of the promoter for RNA polymerase, thereby increasing transcription of the genes downstream from the promoter. The affinity of cap for its binding site on the DNA is enhanced by its association with CAMP, whose level is highest when the amount of glucose in the medium is lowest. Thus, when the inducer lactose or IPTG is present and there is no repressor bound to the operator, a high intracellular concentration of CAMP can lead to a high level of transcription of the genes downstream of the lac promoter. In practice, lac UB5, a variant of the LAC promoter that contains an altered nucleotide sequence in the -10 region and is a stronger promoter than the wild-type LAC promoter, is used in plasmid expression vectors. The TRP promoter regulates transcription of the genes that are necessary for the biosynthesis of amino acid tryptophan. This strong promoter is negatively regulated, turned off, but the TRP repressor protein complex with tryptophan, which binds to the TRP operator and prevents transcription of the TRP protein. De-repression turning on of the TRP promoter is achieved either by removing tryptophan or by adding 3-indole acrylic acid to the growth medium. Unfortunately, repression of this promoter is not very efficient. It is leaky, which leads to a continuous low level of transcription even when the gene should be turned off. Because of this promoter operator cannot be used to express genes that might be toxic or otherwise deleterious to the growth of E. coli. The TAC and the TRC promoters are commonly used hybrid constructs that are similar to one another, different by only a single base pair. Both promoters contain the 35 region from the TRP promoter and the 10 region from the LAC promoter, separated by either 16 TAC or 17 TRC base pairs. BP, both of the promoters are repressed by the LAC repressor and can be induced in the same way as a LAC promoter by the addition of lactose or IPTG to the medium. It has been estimated that the TAC and TRC promoters are 3 times as strong as a TRP promoter and 10 times more effective than the LAC promoter. The P1 promoter is controlled by the CL repressor protein of bacteriophage lambda. In practice, a temperature-sensitive mutant of the CL repressor C1857 is generally used to regulate P1-directed transcription. Cells carrying the temperature-sensitive CL repressor are first grown at 28 to 30 degrees centigrade, a temperature at which the CL repressor prevents transcription directed by the P1 promoter. When the cell culture reaches the destined stage of growth, often the mid-log phase, the temperature is shifted to 42 degrees centigrade. At this temperature, the thermosensitive CL repressor is inactivated and transcription can proceed.